Chapter 3, Microbial Growth. Microbial growth is an increase in the population of microbes. That means the increase in the number of cells rather than an increase in size. So usually when we say someone's growing, we're looking at how they're growing in size. But in microbiology, we look at the increase in the number of cells. The requirements for microbial growth can be broken down into two categories, physical and chemical. In this first part, we'll be covering the physical, temperature, pH, osmotic pressure. In the second part, we'll be covering the chemical requirements, which is carbon, nitrogen, sulfur, phosphorus, trace elements, oxygen, and organic growth factors. Temperature is one of the most important environmental factors affecting growth and survival of microorganisms. At either too hot or too cold a temperature, microorganisms will not be able to grow. Cardinal temperatures are temperature ranges. Cardinal temperatures of different microorganisms differ widely. Cardinal temperatures are three different types of temperatures. The first one is called the minimum growth temperature. Minimum growth temperature is the lowest temperature at which an organism will grow. Below this temperature, the cytoplasm freezes, so it no longer functions in nutrient transport. So the cytoplasm is going to freeze, so nutrients can't move back and forth. So the organism stops growing. Optimum growth temperature is the temperature at which an organism grows best, at which it grows most rapidly. As the temperature rises, chemical and enzymatic reactions in the cell proceed at more rapid rates and growth becomes faster. Maximum growth temperature is the temperature or the highest temperature at which an organism will grow. Above this temperature will lead to protein denaturation, membrane collapse, and lysis. That means that the organism will die. So here's a picture showing you the growth. So here's temperature and then the growth rate. So as it's going, you can see it's growing and growing and growing, and then once it hits its maximum, it dies off. Temperature classes. Psychrophiles. Psychrophiles are cold lovers that have low optimal temperatures. Their optimal temperature is about 10 to 15 degrees Celsius. The range is negative 10 degrees Celsius to 20 degrees Celsius. They're usually found in constantly cold environments, so mostly found in algae and sea ice and snowfields. Psychotrophs, also known as facultative psychrophiles. They grow between 0 degrees Celsius and 30 degrees Celsius. Their optimal growth temperature is 20 to 25 degrees Celsius. So they can grow best at refrigerated temperatures, but can grow at low temperatures. They're found in soils, water, and foods in the fridge, and they can cause food spoilage in refrigerators. They do not cause disease in humans because they cannot survive at body temperatures. So this is a picture showing you the temperature ranges in which certain organisms or psychotrophs will grow. Mesophiles are moderate temperature lovers that have mid-range optimal temperatures. So optimal temperature of 36 to 38 degrees Celsius, so the body's temperature. They're usually found in warm-blooded animals and aquatic environments. It's very common. E. coli is a typical mesophile, so human pathogens are mesophiles. Thermophiles are heat lovers. They have high optimal temperatures, temperature at which water will cause pain if you were to immerse your hand. So if you were to take your hand and put it in 58 to 65 degrees Celsius, it would be very painful. It's usually found in warm environments, so about 40 to 73 degrees Celsius. So compost, soils, hot water heaters, and some hot springs. Hyperthermophiles have very high optimal temperatures, so 90 to 100 degrees Celsius. Their range is about 65 to 120 degrees Celsius. They have very high optimal temperatures, and again, can be found in usually hot habitats such as hot springs, geothermal vents, volcanoes. Mostly archaea, so usually most archaeas are hyperthermophiles. Thermophiles and hyperthermophiles do not cause disease because they freeze at body temperature. So here's another picture showing you temperature versus the rate of growth. So different temperatures in the different classes. pH. Organisms have ranges of acidity that they prefer and can tolerate. Most bacteria grow between a pH of 6.5 and 7.5. Fungi, as a group, tend to be more acid tolerant than bacteria. So molds and yeast grow between pH of 5 and 6. Bacterial growth can lead to a change in pH due to metabolic reaction. Many organisms produce acidic waste products that accumulate in their environment until eventually they inhibit further growth. Chemicals called buffers are frequently added to microbial culture media to make the pH relatively constant. Two classes within the pH ranges. Acidophiles are acid lovers. They grow in acidic environments. So fungi in general and some obligate bacteria. So we see this in Theobacillus and acid mine drainage. They grow at a pH of 1. Aquaphiles are base lovers, and they grow at a very high pH, usually more than 10 to 11, and you can find them in soda lakes and high carbonate soils. Back in chapter 2, we talked about osmosis, and we said that osmosis was when water moves from a region of high water concentration, so to a region of low water concentration. So it's going from low solute concentration to high solute concentration. For typical cells, the cytoplasm has a higher salt concentration than the environment, leading to what we call positive water balance. Remember in chapter 2, we talked about a hypertonic environment. We said that hypertonic environment causes plasmolysis, which causes cell growth to stop due to the plasma membrane pulling away from the cell wall and the volume of the cytoplasm shrinking. So this is usually caused by a hypertonic environment. So here's a normal solution, and here's a solution that's very hypertonic. So notice the dark blue. And this will happen if we were to increase salt or increase sugar. And again, you'll see the plasma membrane pulling away from the cell wall. Remember, the cell wall remains intact, okay? And the volume of the cytoplasm is going to shrink. This is helpful when using salt as a preservative by inhibiting bacterial growth by drawing water out of any microbial cells that are present, preventing the growth and reproduction of microbes. So it's good to use salt because then salt prevents them from growing. Halophiles are salt lovers, and they can withstand a high salt concentration. Extreme or obligate halophiles require high salt concentration, usually 30%. Facultative halophiles tolerate salt. They don't require high salt, but they can survive at high salt concentrations at about 2 to 15%. Here ends part A of chapter 3.